I'm Troy Sandberg, and today I'm going to be talking about a study that I performed in large part as a PhD student in the Systems Biology Research Group under Professor Bernard Paulson. So the motivation behind this study was to examine at a molecular level how a cell differentiates between self and non-self components, because genes are shared across species all over the tree of life. But does that mean you can take a gene from one species and put it in another and expect it to function? And this is not simply an academic question. It has important implications for how life evolved, because horizontal gene transfer happens in the wild all the time, where DNA jumps between species. And it's important for engineering of organisms, because now we can use CRISPR to manipulate microbes or other species and use them to produce valuable chemicals or have some sort of phenotype that we want them to have. So a way to address this question is with evolution. And so in this study, we took a number of species from a close bacterial relative of E. coli's to a more distant relative to an extremophile and to humans. And we took their DNA and we used it to replace a couple of E. coli's important native genes. These are genes involved in breaking down sugar. And if you remove them entirely, the E. coli grows about 5 times more slowly. So there's a big incentive for them to figure out how to use this foreign DNA if they can't use it at first. So once we generated these gene swap strains, with the exception of using donor DNA from the close species relative, E. coli could not use the foreign genes. They grew as if the genes had been removed entirely. And so then what we did is we threw them on the evolution machines, which are things I helped develop as part of my PhD. And they're automated systems that let us generate these fitness trajectories, as you see, that let us track in real time as the populations are evolving. You can see when a mutant arises, it takes over the population, the growth rate jumps, and this process continues. And this allows us to go in and grab these mutant strains and sequence their genomes to see what mutations they acquired. And you can see that there were different outcomes. Sometimes the strains evolved properly and figured out how to use the foreign DNA and reached a high growth rate, whereas in other cases, they were unable to get the foreign gene working and they were stuck in a lower growth rate plateau. And this is because evolution is a stochastic process, so you repeat the same process and you can get different outcomes. So when we looked at the mutations these strains were acquiring, it made perfect sense. Every single case where the strains reached a high growth rate, they had mutations in or around the foreign DNA. Whereas in every case where they were in the lower growth rate and we assumed they had not figured out how to get the gene working, they did not have any mutations in or around the gene. So what were these mutations doing? Well, the paper gets into it in more depth, but basically they were just aimed at increasing the expression level of these genes. The genes weren't functioning as well as E. coli's native copies, but provided you had enough copies of them in the cell, it was sufficient for the protein to carry enough flux to make E. coli happy and let them eat sugar happily and grow fast. Surprisingly, the actual DNA itself didn't need to mutate for the most part. It was perfectly fine, and the cell could grow very happily as long as it got upstream mutations or regulatory mutations that increased the expression level, even though the DNA was foreign to it. So the impact of this work is that it establishes the influence of a number of DNA and protein sequence particulars on interchangeability across species. So genes are interchanging across species all the time in the wild through horizontal gene transfer. And here we're watching this happen in the laboratory in real time as DNA is evolving functionality in a new host. And so the mutations we're seeing here can be used in genetic clock studies where you look at different species and how they've differed over time and sort of rewind time by undoing those mutations to estimate when they diverged from each other. And it's also important for the engineering of organisms, because humans like to make organisms do things we want them to. Like before we had bioengineering, the way you got insulin for diabetics was to filter pig's blood and use pig insulin. And now we just pop the human gene for insulin into bacteria and feed them sugar and get all the insulin we want. And so going forward, we're going to be able to do more and more crazy types of engineering designer organisms. And the mutations we found in this study help point the way towards the optimal way to do that.